have a nice team of an analytical chemist and a pharmacist doing research at Erasmus Medical Center.
Yeah, first off, thanks for having us. My name is Stef Schouwenburg. I'm a pharmacist and a PhD student. And I'm currently conducting a clinical trial at the Erasmus MC about beta-lactams in the pediatric intensive care unit. And today I'm joined by Soma, my colleague. She's sitting next to me.
Hi everyone, I'm Soma Bahmany and I'm working as a research technician and PhD student at the Hospital Pharmacy Laboratory in the Erasmus MC. And today I'm going to tell you a little bit more about the use of more alternative and patient-friendly sampling methods for the quantification of drugs.
Yeah, so today we're going to try and give you a small sense of all the alternative sampling methods and microsampling methods we are using in Rotterdam. Our situation in Rotterdam is certainly not perfect, but we'd like to give you a grasp of what's happening here, and I hope to inspire you maybe to try and pursue something with these sampling strategies yourself. So first off, I'm going to talk for the first 10 minutes about a small review we did about scavenged sampling. If you look at sampling, you could say there's a way of sampling named sustainable sampling, in which you try to use less materials and try to take less blood volume from the patients, and you can subdivide it in microsampling, of which there's dry blood spots, of which Soma will tell a little bit more. Volumetric absorptive microsampling, which is kind of the same as dry blood spots, but yeah, you dry blood on a matrix and then you extract it. Then there's solid phase microextraction, where you use an extraction phase to take diluted blood or urine or any matrix from a buffer, and then try to quantify drug concentrations. And lastly, capillary microsampling, where you make use of capillaries. Then there's sparse sampling. So sparse sampling is, so to say, you take a small number of samples from a large number of patients. So that gives you a bit more flexibility in sampling times, especially when you're doing POP-PK studies. And it's not comparable to dense sampling, of course, because you're taking way less samples from your concentration time curve. And then lastly, there's scavenged sampling. Scavenged sampling encompasses like the use of all residual material left over from standard of care. So it could be blood, urine, liquor, or saliva. And there's some great benefits about using scavenged samples, as there is no extra burden on patients. There's less pricks required for research, and a lot of people experience anxiety due to these pricks. And a great benefit of having less research procedures, that there's a reduction in research materials, as well as the strain on clinical personnel, of which you may all know that research procedures are always extra above on top of clinical procedures. So that's a nice benefit as well. So first off, if you look at the applicability of scavenged samplings, you need to determine what kind of population may benefit from the use of scavenged samples. So especially in pediatrics, where according to the WHO, you may only sample 3% blood volume during a 4-week period, or 1% blood volume at a time. Especially in pediatrics, kids may benefit from using scavenged samples left over from standard of care. Then there's the elderly and the pregnant. In pregnant, you have, of course, washout studies, for which it's very important. And then lastly, if you look at the ICU setting, there's lots of residual material which is currently not used, and may have great potential. Then you need to determine what's your purpose. Do you want to quantify drugs for TDM? Do you want to look at biomarkers for disease progression, toxicity, or antibiotic resistance? And lastly, you could even determine genetic material. You could look at the metabolism of drugs due to polymorphisms. Yeah, then there's of course the research purpose or the clinical purpose. Drug quantification can maybe used for TDM or modeling for precision dosing, as well as continuous monitoring, of which I'll tell a little bit later. How to realize a situation like this? It's really important to go to the ward, see how all the logistics around blood sampling are currently happening, so you can identify the hurdles and barriers for the implementation of scavenged samples. It's really important to talk with the clinical personnel, because they actually know what's happening, and educate them. You see, by educating them, you create a certain understanding why you're doing this, and why it's so important to register sample times. And by being actually at the ward, you try to create visibility and accessibility. So when there's questions or when something's going wrong, it's easy to access you. Now I'd like to look at the opportunities for scavenged sampling. So if you look at strategy development, at the sampling times, so to say, you're dependent on standard of care sampling times. Scavenged samples may have a little less volume than you're used to. So especially for antibiotics like ceftriaxone, where you're trying to determine the free fraction, it may not be feasible because you don't have enough material for your assay. As well as you need to be aware that there may be an underestimation of the drug quantification in your samples, especially when you're doing POPI case studies or anything like that. It's really really necessary to understand what kind of variability you're introducing into your data set. But ultimately, if you're collecting more data via scavenging, it may be enrichment for your data set. So as I said, they're scheduled blood draws, so you can't really determine when scavenged samples are being taken. But the huge downside of this is there are documentation errors, and I think that many people who do POPI case studies are encountering something like this. You're not quite sure whether a certain drug is given at the right time, or a sample is taken at the right time, which is registered. So it's really really necessary to be on top of that. And here there might be a huge opportunity for automation. So to say, you're trying to digitalize every step in your process so that there's less chances of anything going wrong. The good thing about scavenging is that you're kind of creating a standby infrastructure. So as once you've implemented at a certain ward or a department, it's very easy to turn it on and off again. So if you train your clinical personnel, there's lots of ways how to use residual material, but something to be aware of is of course sample instability. You're not sure how long after a sample is taken it's taken to the cooler or to the freezer. And the important thing to realize is that for certain drugs, like freeze-thaw cycles may compromise the stability of the drug. So it's really important to have a multidisciplinary approach, close contact with all the involved. Lastly, there's the analysis. So what you're doing is a method comparison. At first, you want to validate your matrix, so you compare it to actually scheduled samples. A downside of this is that there is an increase of performed assay, but the potential opportunity for the lab is try to develop multi-quantifying assays. So if you look at the literature, we found out that there's a lot of studies being done in kids with blood, but urine, saliva, mucus tissue, or liquor is barely used as residual material. As well as there's one article being published about the use of residual material for pharmacogenomic testing for the OCT1 ontology is used for morphine. If you really look at the future, so you might be able to establish a delayed real time due to scavenged sampling. Delayed because you're not measuring in real time, but it could give you a really good sense of the concentration time curve for certain drugs or biomarkers, which are really essential for disease progression or circadian rhythm or anything like that. And then you might be able to use prediction models for biomarker-guided infection therapy. Lastly, I'm currently conducting the EXPAT KID study at the Erasmus MC. It's a clinical trial about the effectiveness of beta-lactams in the pediatric intensive care unit. And if you look at this small graph, we're currently taking a trough and a peak sample around a dosage moment. And then the days afterwards, we're collecting S-strip syringes. At the same time, our PK sample is collected. So we can compare the use of S-strip syringes with our PK samples. And it's important to validate because there's heparin in the S-strip syringes, which may compromise your sample stability. So we're currently still performing this research. Now Soma is going to take over and she's going to tell a bit more about the laboratory in Erasmus MC.
Yeah, so first I would like to explain a little bit more about our mostly used quantification technique in our laboratory, which is the LC-MSMS. The LC-MSMS is a commonly used analytical technique in laboratories, which stands for liquid chromatography coupled by mass spectrometry. And first, at the chromatographic part, compounds are separated based on their polarity. And after the chromatographic separation, compounds are separated in the MS by their mass over charge ratio. So the MS provides a selective detection, and the combination of LC with MSMS results in highly accurate, selective and sensitive quantification technique, which is of crucial importance, especially for the TDM of neonates, because due to the increased sensitivity, lower sample volumes are required. So there are different ways to make your quantification method more sustainable and also more patient-friendly. One of them is the development of multi-methods, because that enables the simultaneous quantification of different drugs in one method, which will result in lower required sample volumes. And also another option is to make use of more patient-friendly sampling methods instead of the standard vena puncture sampling method. So to get the most out of one sample, it's very important to make use of multi-methods, as I explained before. Here I have some examples of the multi-methods we are using at this moment in our laboratory. And the simultaneous quantification of drugs results in lower required sample volumes, but also in faster TDM and toxicological advices. Although it might be sometimes challenging to combine all these drugs in one method because of their different chemical properties. So now I would like to explain a little bit more about the difficulties with regard to the method development of multi-methods. There are several aspects that may have influence on the chromatographic settings and the MS settings. For example, every compound has a different signal in the MS. That's why the sensitivity of every compound is different. Also, the used quantification method and detection method is very important. And also the chemical properties of every compound is different. For example, some compounds are more polar or more non-polar, and that's why they differently in the LC. And also the therapeutic range of the drugs is important. Some drugs may be analyzed in much higher concentrations compared to other drugs. So all these aspects are of influence. Nowadays, more alternative and patient-friendly sampling methods are upcoming. One of them is the dry blood spot method, DBS. DBS gives the patient the opportunity to perform their blood sampling at home using a finger prick, and after drying the DBS card, can be sent to the hospital by mail. And next to DBS, there's also another alternative for blood sampling, which is saliva. Sample collection can be performed using a swab, which is long enough to hold one end while the other end is placed in the mouth. And not the only medical center in the Netherlands which is looking for more alternative sampling methods. Also in the medical center in Amsterdam published a paper of the validation of gentamicin in saliva for TDM, and they compared the concentrations of gentamicin in plasma with saliva, and those concentrations were comparable with each other, which is also very hopeful for development of other drugs in saliva. So now we have some experience with the DBS method. That's why I would like to share the advantages and disadvantages of the sampling method. One of the advantages is that small sample volumes are required. It's also less painful and less invasive for the patient. The patient can perform their blood sampling at home, which also results in less hospital visits. Although DBS has a lot of advantages, during implementation in clinical practice, we observed also some difficulties. We saw that it's very important to give the patient the right instructions, because there is a high risk of making mistakes during blood sampling. Also, the transport time and transport conditions may affect the concentrations of the drugs on the DBS cards. Also, during the measurement of the patient samples, we observed some deviations, which may be caused by the hematocrit, because the hematocrit has an effect on the spreading of the blood spot on the DBS card. Also, it's difficult to share your sample with other laboratories, especially when there is only one blood spot on the DBS card. And also the extraction of the drugs on the DBS spot is lower compared to the extraction of blood. And that's why it makes it more difficult to measure the lower concentrations. So for example, when a blood spot is too small, there will be not enough material to perform sample analysis. But also if there are multiple drops in a blood spot, it may also affect the measured concentrations. Furthermore, patients should avoid to touch the DBS card to prevent contamination, and patients can not squeeze their fingertips to get more blood out of it, because that will dilute the blood, and all these mistakes are not always visible on the DBS card, but they could lead to a bias in the measured concentrations. So after the patient has performed their blood sampling, the DBS card should be sent to the hospital by mail. But when implementing DBS, one should consider the time that it will take to reach the laboratory, as well as the sample stability during transport and in the transport conditions. Also, the transport time has consequences for the moment of sampling, because if the doctor wants to check laboratory results before the patient visits the hospital, and it takes a few days to reach the laboratory, then the blood sampling must be performed 1 week before the appointment. And moreover, the sample should be stable over time, because if the analytes decreases on the DBS card, then the measured DBS concentrations are not comparable to the real concentrations. So we should be aware of all influences. But what's next? Now we have developed and validated methods using the near infrared spectroscopy to measure the hematocrit levels on DBS to correct for this hematocrit effect. Also, we are making videos to give the patient the right instructions and training for the DBS blood sampling. And also we are trying to optimize our methods to validate them in lower sample volumes.
Yes, and to wrap things up, we have a little recap of our presentation. Yeah, when considering to use microsampling or scavenged sampling, it's really beneficial to know what the goal is of your actual research. So whether you're trying to make a pop PK model and want very narrow estimates of your PK parameters, scavenged sampling might not be the best option for you, because you won't be able to get a very dense sampling. If you want an estimate of your PK parameters, it might be a solution. It's very important to have very good communication with all the departments involved. As to say, if you go to the laboratory and talk to your analyst, it's very essential to know what kind of assay is currently available. When using microsampling, you're using less volume, so you need to be certain that what your actual plan is at the ward may be feasible for the lab as well. And lastly, consider sample stability and storage conditions. When you're using new matrices, there's always new factors that may compromise your compound stability, and therefore you should always take into account all the logistics around sample storage and the storage conditions. Yeah, we hope we've been able to give you some new information about scavenged samples or microsampling, and we're very happy to answer any questions. And thanks so much for your attention.
Thank you very much, Stef and Soma, and thank you very much for taking the time to work through the problems that we're encountering.